Hello everyone, my name is Arnav, and it is my pleasure to present the BENG 168 video series by the Big Bang. BENG 168 is about biomolecular engineering, and in this series of videos we'll be going over several biotech techniques that are very applicable to this day. In this video we'll be going over details on restriction enzymes and plasmids. What are restriction enzymes? Type 2 restriction enzyme nucleases, also known as restriction enzymes, are molecular scissors that cut DNA molecules internally at specific base pair sequences. These scissors look for a short sequence known as a recognition site. These sites can range from 4 to 8 base pairs and sometimes even more than 8 base pairs. These scissors can make two types of cuts, a straight cut and a staggered cut. For the straight cut, the scissors cut the two DNA strands at the same base pair position. As a result, blunt ends are formed. In the staggered cut, the scissors cut the two DNA strands at different positions. This cut creates an overhang known as a sticky end, because it is a lone strand which can combine with another DNA strand. So we covered restriction enzymes. Now let's talk about plasmids. Plasmids are self-replicating, double-stranded circular DNA molecules maintained in the bacteria outside of the chromosome. For self-replication to occur, each plasmid usually has a sequence that serves as its own origin of replication. That way it can replicate independently of the bacterial chromosome. The main advantage of plasmids is their ability to self-replicate. This feature enables convenient transfer of DNA between different bacterial cells. In turn, plasmids can serve as vectors or delivery vehicles to carry cloned DNA. Another important feature of plasmids enabling them to be cloning vectors is the multiple cloning site, or the MCS. The MCS contains several unique restriction enzyme sites which allow DNA fragments to be inserted in that region. Now let's take a look at an example of a plasmid cloning vector. A commonly used plasmid cloning vector is PBR322. So in this plasmid we have two antibiotic resistance genes, one for ampicillin, one for tetracycline. We also have the origin of replication, of course, and we have several restriction enzyme sites. Let's take a look at one in particular, the EcoR1 site. EcoR1 will make a staggered cut, and this will be important later, as you'll see. So first thing, to use this as a cloning vector, we want to cut the plasmid using EcoR1 enzyme. Then it will become a linear piece of DNA. Once this DNA is linearized, we will insert the gene of interest. The key thing for this gene of interest is there should be EcoR1 sites that flank it. This is because, as I mentioned with the EcoR1, it creates a staggered cut. So there will be little overhangs at the end which will enable this gene of interest to be integrated into the linear piece of DNA. Once this gene of interest is integrated, then we will circularize the linear piece of DNA using T4 ligase to create a new plasmid with our gene of interest inserted. We have seen how to create a cloning vector, but what is the process to insert that vector into bacteria? Transformation is the process of taking in foreign DNA. Cells that are capable of taking up foreign DNA are known as competent. The process of transformation entails the binding of double-stranded DNA to components of the cell wall, entry of DNA into an inner compartment where it's protected from nucleases, and transmission of one strand into the cytoplasm while the other strand is degraded. If DNA is linear, integration into the host chromosome. If it's a plasmid, it's maintained in the cytoplasm after the second strand is synthesized. Competence is usually not an intrinsic property of bacteria. It can be introduced through chemical means such as cold calcium chloride and a heat shock, or electrical means such as a short electric shock. After transformation, one needs a screening method to ensure that the cell successfully took the DNA. For example, after a PBR322 transformation, cells are incubated in a medium without antibiotics, as you can see with the flask. Then the mixture is plated onto a medium containing ampicillin, or a plate containing ampicillin. The cells that survived are transferred to a plate with tetracycline. The cells that survive the tetracycline plates have both the ampicillin and tetracycline resistance genes, thus they successfully took the PBr322 plasmid. To summarize, in this video, we discussed restriction enzymes, their applications, plasmids, and how plasmids are used as cloning vectors. Here is our source. We hope this was helpful, and thank you very much for watching.